Hi all, welcome back. We're going to continue talking about the nervous system. And this time out, we're going to talk about the different components of the nervous system, like what kind of cells make up the nervous system, what do those cells look like, how do they act, what's, what's the job of the different components of the cell, etc. So first off, nervous tissue is made up of two main types of cells. You have neurons and neuroglia cells. The neuron can vary in size and shape, and this is really when we think of the nervous system, we're thinking of neurons because these are, or this is the site where action potential, this is a site where signals are sent from one neuron to another, or a neuron to a muscle cell, or a neuron to a gland. And this is the site of signaling. Okay. Um, so length, etc., can definitely change. We often use neurons as an example of atypical cell. Neurons can be fairly large, and they can be fairly large because of dendrites, which are these tentacle-like projections that come off the neural body. That increases the surface area of the neuron, which allows for them to be larger than, say, a red blood cell, or even a white blood cell, or most other cells. Okay, you have some cells, like we talked about before when we were talking about muscle cells, like skeletal muscle cells, that can also be very large. They get around that surface area problem by increasing the number of central command centers. So they have multiple nuclei per cell. In this case, single nucleus, but projections which allow for the surface area to increase. Okay, so first of all, they have a cell body. Sometimes this is called the soma or the perikaryon. Okay, it contains that nucleus, one nucleus, cytoplasm, organelles, etc. We'll talk about some of these substances as we move on. It has dendrites. Okay, so these are branched receptive surfaces. Okay, how many they have really just depends on the type of neuron, and we'll come back and look at this. They have an axon. The axon's purpose is to transmit a signal, and so they do so by releasing neurotransmitters. We've already talked about one type of neurotransmitter when we talked about muscle tissue, acetylcholine. We talked about that quite a bit. Now, there are other potential neurotransmitters, and axons would be associated with those. Okay. Again, what the axon looks like depends on the type of nervous cell or type of neuron. Okay, and so they normally have one axon, but it can be a split axon, or it can be hard to distinguish what the axon looks like. And so we'll look at this here in a second. So let's go through the axon. This is what they kind of look like. So this is a nerve cell. That's the cell body, the nucleus, the organelles, the dendrites on the outside of the cell body. But one of those regions around the cell body is going to have a longer, different conformation, a different structure, which we call an axon. Sometimes it kind of looks like dendrites, but it's an axon because it's tipped with synaptic knobs. So let's look at the different features. Okay, so first they have a HELOC. Okay, the HELOC is just that connection point between the cell body and the axon. Okay, and there's a distinct amount of neurofibrils that are in that region that kind of distinguish where that HELOC forms. Okay. Collaterals are any branch that comes off the axon, off the main axon. So anytime it branches, that's considered a collateral. Okay. The axon terminal is the end of the axon, okay? And that the axons end with synaptic knobs. Okay, so the axon, when it splits at the end, it's going to have a synaptic knob. That synaptic knob will hold the neurotransmitters that are going to be moved from one cell to another. It could be from a nerve to a nerve. It could be from a nerve to a muscle, a nerve to a glandular cells. But that region right there is what's holding the chemicals that are going to be moved. Okay. If we dive into a little bit of other cells that are associated with the axon itself, you have these specialized cells that cover the axon, which are called Schwann cells. The Schwann cells themselves are made of a couple different things. First of all, it's a cell, so it's going to have its own nucleus, it's going to have its own membrane, its own cytoplasm, but there is an excretion of material called myelin. Myelin is fat and protein, but the important piece and the thing to kind of remember is the fat part really limits what can be transmitted in this region. Okay, so remember fats, they're a barrier typically to things that have charges. Okay, so just like the membrane of a cell, it doesn't allow things that have a charge, which we call ions, it doesn't allow them to move across that membrane freely. Same deal here with the myelin, which makes up what we call a myelin sheath. Okay, so a wrap or a coating that surrounds the axon. Okay, and it really composes of both the Schwann cell and then the myelin material, which protects the axon but also limits what can enter and exit in this region, which is where the nodes of Ranvier come in. The node of Ranvier is a region where there is no Schwann cells and no myelin sheath, so it's bare axon, and those are the sites where ions can move across, right? Which is important because the action potential, which we've talked about a little bit before, the action potential is directly related to how ions change. The polarity of a cell changes over time. Okay, so moving from a negative state to a positive state, back to a negative state. Remember, we talked about these action potentials in this way. All right, not all axons are myelinated, but when they're myelinated, okay, you have this sheathing material that covers the neurofibrils or the axon. Okay, and again, that sheathing material is called myelin sheath, okay, and it's a component of protein and lipids, or protein and fats. Okay, and the nice thing with that is it allows for the impulse, it allows for the electrical impulse to move faster. So it might not allow for things to enter, but things that are already in the system, it allows for them to move faster in that region. So having myelinated sheaths on your axons means that your electrical impulse in that region can move faster. Okay, and that's important. Okay, when we look at the central nervous system, any part of the central nervous system that has myelinated axons, we would consider that white matter, and this will play an important role in a little bit later. If it's non-myelinated, it's considered gray matter. Okay, and oligodendrocytes are what's laying down that myelin material, and again, it's really important. And a lot of immune or autoimmune disorders or diseases are the result of the myelin sheath being destroyed. Either the immune system eats away at it, or in some cases, maybe genetically, the oligodendrocytes are not that good at laying down myelin. Maybe the myelin sheath is thinner. And so you can have different genetic disorders. You can have different diseases that can develop where it's affecting the myelinated regions of the axon, which affects the speed. And it also, if you remove it, it allows things to enter, okay, which can change the impulse and may disrupt the signal altogether. Unmyelinated regions, okay, this occurs in the central nervous system also. So here you can see myelin sheath. Okay, if we did a cross section, you can see this dark matter, okay, is this myelinated sheath. Okay, the axon's here running through. Okay, if it's unmyelinated, you can see it's just the axon. There's a little bit of membrane around that axon, but it's just the axon. There no bands of myelin that go around. Still Schwann cell associated with it, okay, on this outside, but again, no myelination. Okay, so one of those disorders, one of those diseases that I was talking about where you have damage to the nervous system is multiple sclerosis, okay, or MS. Multiple sclerosis is when the myelin sheath in the central nervous system is destroyed, okay, and it's normally destroyed by your immune system or the immune system of the individual that has MS. Okay, so your own immune system is producing antibodies against the myelin. Okay, so the myelin is being destroyed, and the sclerosis part, sclerosis means scar. Okay, and so when this happens and the myelin is being deteriorated, new material will be laid down, but the new material is a stopgap. It's to try to cover the axon, but it's not using the same material that myelin's made out of, and it produces a scar. And that scar doesn't protect the axon, and it doesn't allow for the signal to send, okay? And so it really stops that impulse from moving down. So you have this action potential, this wave that's moving down the axon all at one force, but then you have this region where the myelin's not there any longer and it's a scar, and it really stops that impulse, okay? Or deadens that impulse so it doesn't reach or slows down quite a bit, okay? When this occurs to a great deal, okay, then you know, just like we were talking about before when we were talking about skeletal muscles, if you don't use the skeletal muscle, then it will go through atrophy. Okay? In this case, atrophy can occur when that motor neuron, or when that motor, when the motor neuron doesn't reach the muscle or doesn't reach the region where that neuron would terminate, okay, then the impulse never triggers neurotransmitters to be released. You don't have any calcium gates, you don't have any sodium potassium channels opening up, and you don't have the muscle contracting. And so another way you can get atrophy is by not having a nerve impulse reach the muscle itself. Okay, this can cause lots of problems. It occurs not just in the muscles that are used to move the body, but the muscles that are used to do things like see. Okay, it can cause fatigue in certain regions because some muscles are now overactive. some muscles have to make up for the regions that there is no neurotransmit into that region, so other motor neurons have to kick in and they're overactive, and then they become fatigued because there's not enough energy, there's not enough recovery time, and you can get fatigue, mood problems, blurry vision, all kinds of things. Obviously, weak or problems in the limbs because a lot of the motor neurons are going to occur in that region. Lots of treatments have been studied and brought to the forefront for MS, but most of them just are trying to make it so your immune system is less active. So they suppress the immune activity, which we know can be problematic. And not only is the immune system protecting your body from, viruses and bacteria and things like that, it also helps with destroying cells that might be damaged, cancerous cells, these kind of things. So if you suppress your immune activity to help stop the motor neurons from being damaged, then you can increase the likelihood of illness, sickness, even cancer from developing or to develop. All right, let's look at some classifications real quick. So you can have a neuron which is that typical shape and structure that I showed you before, where you have dendrites, many of them coming off the body, and one axon. This is called a multipolar neuron, okay? And this is the majority of the neurons in your body. Okay? Most of the neurons in the central nervous system and some in the peripheral nervous system. Okay? Bipolar neurons, this is when you have one dendrite and one axon. It looks like there might be two axons, except for, you remember, the axon's going to have a terminal end, and it's going to end with that neurotransmitter region. Okay, so you know that only one end has neurotransmitters. Okay, not super common, but it is found in the eyes, ears, and nose. Okay, unipolar or pseudo unipolar. Okay, this looks like it has a single axon. Okay, and no real dendrites, and it's split. So it looks like the axon is branched or split, and has two branches. And this is really important for sending signals in the peripheral nervous system, and we'll talk more about that system in a little bit. Okay, so let's look at them. Again, you have that normal shaped nerve that most people are familiar with, which we call multipolar. Okay, you have dendrites coming off the body. One region is going to have that axon. This is going to be the site of action potential moving through the axon and then across the synaptic cleft, wherever that be attached to. It could be attached to another nerve, could be attached to a muscle, a gland, etc. Okay, the bipolar, the dendrites are coming off in a linear form, not a branch form, off the entire body. Axon comes off on the other side, so this is why it's called polar. So one side is dendrites, the other side is axon. Okay, and then the unipolar is going to have the cell body and then a branched axon. Again, one of the ends is going to be dendrites, the other end is going to be the synaptic region. So, but they originate on the same axon. The same axon feeds both dendrite and synaptic region. Okay, so looking at neurons by their function, we already briefly talked about this last lecture. Okay, you have afferent neurons. These carry impulses to the central nervous system. Okay, so from the peripheral to the central. So these are what you're often going to find in the spine and the brain. Okay, lots of unipolar, bipolar. Um, so let me back that up. You're going to find them on the outskirts of the brain and the spinal cord that are associated with the peripheral nervous system. Okay, so these are the signals that are coming from the peripheral, okay, to the central nervous system. Within the central nervous system, you're going to find interneurons. Okay, these are link neurons. Okay, or sometimes it's called the association of neurons. This is where all the neurons, and I think probably some of you have seen videos of all the neurons in a person's brain firing and connecting and things like that. Those are interneurons. Okay, those are multipolar bodies. Okay, that typical nervous system, lots of glia associated with them, and relay information from central nervous system to another part of the central nervous system. So you have afferent moving from the PNS, or the peripheral nervous system, to the central. Okay, when the signal gets into the central, okay, you have interneurons that move the signal between or different parts of the central nervous system. So from like the spinal cord to the brain back to the spinal cord, within the brain, etc. And then you have motor or efferent neurons that are moving that impulse from the central nervous system back to an effector, a muscle or a gland, or both. They're also typically multipolar. Okay, and we already kind of talked about, in some cases you have the voluntary control, so if it's going to a skeletal muscle, and then other cases you have involuntary control if it's moving to smooth or cardiac muscle or glands. Okay, so just looking at that pathway again. Peripheral nervous system, you're going to have a sensory receptor. Okay, this is normally a unipolar nerve. Okay, that it's going to end in a receptor, and that signal will be transmitted through the axon. The axon will be attached to another region, typically closer to the spinal cord, depending on where this is occurring. Okay, that signal will be sent to a multipolar nerve. That multipolar will send another signal or multiple signals throughout the brain. Okay, a response will occur. That material gets sent back to the spinal cord and then sent to a motor neuron, which is going to connect with an effector, a muscle or gland, etc. We'll look at this in much greater detail later on. Okay. Also associated with the nervous system is that other group of cells, the neuroglia cells. Okay, so you have these neuron cells where you have these dendrites and an axon. Okay, but then you have these little teeny cells that are associated with it, much smaller, no dendrites, not branched like that, but they form a connective system sometimes. They form some structure, they provide nutrients, okay? They clean, they take away waste, they can allow for synapses between uh, two neurons to occur more readily. Okay? They can provide ions that are needed in the system, lots of different functions. Okay, so lots of ways at which this occurs. The first way is in an embryo. Okay, these neuroglia cells often will like usher or guide the neuron to the position where the neuron needs to be, and we believe they also probably play a fairly large role in specializing the nerve. So determining what that nerve is going to be and what that region is going to stimulate. So if it's a motor neuron, what is it going to be connected to? That kind of stuff. Okay. The other thing that they do is they produce growth factors, allowing the neuron itself to be nourished. They remove waste, they remove ions, okay, and excess neurotransmitters. So you have a bunch of acetylcholine in the system, okay, they'll help with the removal of that. And then again, they can aid in the formation of synapses between two nerves. Okay, there are a few different types of neuroglial cells, and all of which will have different actions, different kind of purposes. Okay, the astrocytes, okay, these are sometimes called the star neuroglia cells. They form scar tissue. Okay, they aid in metabolism. They regulate ions, and they make up part of that blood-brain barrier. So lots of connection with blood. They exchange nutrients, they exchange waste, uh, put growth factors into the system so the neurons themselves can grow when they need to. Okay, oligodendrocytes, we already talked about them. They're laying down myelin for the myelinated sheaths that cover the central nervous axon. Microglia, these are phagocytic. Okay, we already talked about phagocytes before. Remember, these are the cells that can engulf, bring in material. Okay, so they will often bring in waste or kind of protect the nerve, but they can also provide structural support. And epithelial cells, these are what's providing the resource that's needed to produce spinal fluid. Okay, so the fluid that's associated with the spine is produced by epithelial cells. Okay, and so like when you get an epidermal, that's where that term comes from. Okay, it goes into the spinal fluid. Okay, so really, yeah, important for the control of ions and the movement of action potentials and things like that. We need that fluid in that region. Okay, we can look at some of these. So neuron, okay, oligodendrocytes, excuse me, they're laying down that myelin. So you can see here they're kind of attached with the myelin sheath, and they're direct association with the Schwann cells. Okay, the microglia, they're going to be floating around, capturing things, consuming it if it's, waste material, etc. Phagocytize it. The astrocytes, they're providing the nutrients, they're providing that connection between blood capillaries to the neurons, okay, the growth factors, etc. And the epithelial cells, they're providing the fluid for the region. Okay, they're often have microcilia, okay, so these little hair-like projections that allows for fluid to be moved and pushed and generated via glands or via capsules that are associated with it. If we kind of take a look back, and we already talked about Schwann cells, but there's another group of cells that are associated often with the neural body, and we typically call those satellite cells. And their purpose is really to provide protection and nutrients and ion balance with the body of the neuron. Okay, so astrocytes might be part of that satellite cell, but there's some other cells that we're not going to get into that are also associated with that region. All right, so if we look at the neuroglia of the peripheral nervous system, again, Schwann cells, already talked about them before. Okay, they have that myelin sheath that's associated with them. This is protection, but also allows for the speed of a impulse to increase. So myelinated nerves will move much faster than non-myelinated nerves. Okay, and then satellite cells, again, You got these cells on the outside that are going to provide that nourishment, ionic balance, and protection. All right. The last kind of topic I want to talk about is what happens when your neurons are damaged, destroyed, etc. Even if there's just minor damage. Unfortunately for our neurons, they don't go through the cell cycle, so they don't replicate. Now, there is some nerve cells that can regenerate portions of the cell itself. But for the most part, if the body of the nerve is injured, okay, that neuron will die. Okay, and so neurons in general and the peripheral nervous system, they may regenerate an axon. It depends on where the damage is done and how much damage has been done. Okay, so a couple ways at which this occurs. If there's a tear in the axon away from the body, okay, then it's possible with the help of Schwann cells, okay, and a material called the neuroglia, that there will be a little kind of tube that connects the material, and you can remyelinate that region as long as the connection point is still there. Okay, so the remaining Schwann cells will provide kind of a guiding sheath for the growing axon. That axon can grow back, okay, as long as the Schwann cells are there. And I'll show you a description of this, kind of a time scale of this. If it can return to the same connection point, then function will return. If it's not the same connection point, so let's say it's connected to a gland, okay, and that has been severed, that axon's been severed. If the same connection point is not capable, the cell will die and the function will be lost. Okay. In the central nervous system, it's highly unlikely that regeneration will occur, just because oligodendrites do not proliferate. They do not come in and lay down new myelin sheath after an injury. So it's fairly unlikely that the material will regenerate. Now, there are ways at which you could potentially add new cells to the system, like adding stem cells, where you're just getting undifferentiated cells to come into the system and make new connections. That's possible. But having them regenerate or correct the damage is unlikely in both cases. But the peripheral system, it does happen. This is how it kind of happens. So let's say there's a tear in this axon, site of injury. Okay. So then the Schwann cells that are still in that region, okay, will form a tube, okay, a area regeneration tube. That axon, if it follows that tube, it can reconnect to the motor plate. So remember, the muscle has a motor plate. That's where you're going to have that synaptic cleft. That's where you're going to have the axon terminate at the end there. It can reconnect it. If it doesn't reconnect it, then the cell itself will die and you'll lose motor control in that region. But if it does reconnect it, that axon will move through the Schwann cells. The Schwann cells will start growing again, even lay down new myelin sheath, okay, and you'll have that former connection reconnected. Okay, we'll stop there. And we'll talk about some more disorders, diseases, and how they work for the nervous system.